Ladies and gentlemen, welcome to this latest teaching session in the course on technology and the future of medicine. Today is the Promise and Perils of Nanotechnology Part 2, and Dr. Woodside is giving the main lecture. I have a few remarks. Dr. Woodside sent me an interesting timeline from the BBC, which has an extra feature you may not immediately notice of most likely and least likely at each time period, and the odds of how likely or how unlikely it is. And when you scroll down to 2045, the technological singularity in 2045 is 8-to-1 against it happening, according to these people. Of course, you know, what does the British Broadcasting Corporation know about anything? But anyway, I would recommend that you have a look at this. It kind of overlaps with the other timeline site, but the other timeline site has a lot more data, but this has a lot stronger opinions about things, which is quite interesting. And just a reminder of that other site, futuretimeline.net. Okay, I wanted to tell you a little bit about cryonics. And this may be, it's not very exciting to tell you, the least important thing I ever talk about in this course. It's possible that not only is it not working, but it's not important for you to learn about it. But it's something that I happen to know quite a lot about. It's something that most people who are interested in this are not scientists. And so there's a lot of sort of folklore about it, and the idea of freezing yourself when you're about to die, with the idea that you can be brought back to life when medical science finds a cure for whatever was killing you is superficially attractive. And there are people freezing themselves right now. And there are cryonics facilities with these large Dewar tanks with either whole human bodies or just the head. It's a lot cheaper if you just do your head. And people, it's interesting. You might think the people interested in this would be old people, but one of the most vocal people is a guy who I met when he was 16. He's now 17, and he's a strong cryonics advocate. And he goes to all the meetings, and he's trying to get cryonics for his pets as well, not just him. And so what does this have to do with nanotech? Well, the idea is that how are these disease processes in your frozen body going to be fixed? That nanobots will be specially constructed to go in and fix everything before you're rewarmed. So they figure out what the cure is for whatever was killing you. The nanobots go in and fix it at liquid nitrogen temperatures, and then you're rewarmed and you're completely normal and you live another few hundred years. Now, I would say that not only do I not believe that nanobots are completely temperature independent in the sense of going all the way from absolute zero up, but there's probably nothing in the world that is completely temperature independent in that setting. Within a certain phase, within a certain range, there would be things that are not dependent upon temperature. But I would say that basically in a body that's being maintained in liquid nitrogen, there's no way to get the nanobots in, to get them circulating around. It's a very basic problem. So no matter what you think they might be able to do, there's no way to get them in there until you rewarm the patient first. So Anyway, that's the idea. Advocates claim that tissues can be repaired while still frozen. And they complain bitterly about their difficulties with funding. And they claim that it's because people are spooked by the novel prey and by the specter of gray goo, that it's all the nanotech researchers' fault that they can't get funding for cryonics. But actually, I don't think that's true at all. The truth is that it's because of the pseudoscience. People don't generally like to think of themselves as supporting pseudoscience, and that's really what this is. And the view that I have of it, I attended virtually the last cryonics conference. You know, you can attend online, and I'm probably one of the few people who actually listen to all the presentations, and it comes down to something very simple. The largest organ you can freeze and then have it come back and function is a rabbit kidney. Nothing bigger than that have they ever succeeded in freezing and then bringing back as a completely functional thing or organ. And even the rabbit kidney, they couldn't predict when it was going to work, and most of the time it didn't work, but on rare occasion they would have a rabbit whose kidney after rewarming could sustain the life of the rabbit. Still had chronic renal failure, so it wasn't perfect, but it was functional enough to keep the rabbit going. You get these large clefts when you freeze a whole human being, and they're big enough that you're not at the nano scale anymore. I mean, they're measured in like millimeters. You know, there's nothing that nanotech can do to fix a situation where you get these clefts or tears in tissue. So it's not subtle what goes wrong, and that's true even with vitrification and various fancy ways of freezing things. So basically, the people who are getting frozen now, if they're conscious when they're brought back, they're going to have extremely painful wounds, like all over their body. I would imagine it's probably worse than being dead. I don't know. And then, I don't know how much you know about Texas, but in Texas, near San Antonio, there's a small town named Comfort. And Comfort has a lot of farmland and so on. And the cryonics enthusiasts have bought a large parcel of land there, and they're planning to build this huge facility. This was secret in terms of the location and the plans for it until November 2012, when they finally got up the courage to hire architects. They've been drawing sort of pictures of this thing at low mag for many years. Since 2006, they've been talking about building this huge cryonics facility. But in November 2012, they actually began hiring architects to plan it in detail. It'll still be probably many, many years before it's actually created. The final thing to tell you about it, which I think is kind of funny, is there was a mansion on this land when they bought it, and they had big plans for what they were going to do with that mansion, so they're all into freezing things, right? So one winter, the pipes froze and burst and wrecked many rooms of the mansion, which is sort of the ultimate irony. So as I say, I consider this relatively unimportant, but it happens to be something that I know a fair amount about. If you have friends who are deeply into cryonics, I'd be happy to give them psychotherapy. And now I think we'll pass on to Dr. Woodside. Any questions about this? Okay.
So last class we talked a little bit about some of the things you could imagine doing and some of the problems that we have to overcome in actually making these reality. And today I wanted to talk more about what I would call practical nanotechnology. So this is things that we're actually able to do right now. So this will give you sort of a sense of where we are in some of these areas. And of course, I can't cover all of the areas we talked about last class, so I'm going to focus in on two particular areas. One of them is building structures by manipulating individual atoms. So this is, it's sort of at the core notion behind molecular nanotechnology. But I'll show you what we can actually do with this right now. And secondly, I'd like to talk about this, again, a very fundamental notion, is this idea of biology as a template for nanotechnology. And I'll discuss some particular examples from this that are being studied right now. So to start off with manipulating individual atoms. So there's a tool that was developed about 25 years ago now, or maybe even 30 years ago, called the scanning tunneling microscope. And this actually allows you to see individual atoms on a surface. So it's sort of illustrated here. The notion is that you have this very, very sharp tip, and it's essentially got one atom right on the end. And then you make use of one of these properties of quantum mechanics where an electron can actually go from there to the sample. If both of those are metallic, they both conduct. The electron can jump from there to there without going in between. So this is called quantum tunneling. And it ends up being very, very sensitive to the distance in between. So in fact, you can basically keep a constant current and then move this along the surface, and it'll execute little motions. The tip will move to match basically the topography of the surface. So you can take an image of the atoms on the surface. This was given the Nobel Prize 25 years ago. So you can actually take images of surfaces of atoms. So this is a very famous surface. It's the silicon. It's a surface of silicon. It makes this beautiful hexagonal pattern. You can see each of those blobs is a single silicon atom on the surface. Now, as it happens, you can also, instead of just imaging things, you can push the atoms around on the surface. So this is, I don't know if you can see this in these slides because the contrast isn't very good, but there should be little white dots. Can you see this? Not really? Okay. Yeah, it's kind of unfortunate the lights are too bright. But there's little white dots you may be able to see against the black background, and those white dots are individual atoms, and then they've pushed these around with their STM tip, and in this frame you should be able to, you would be able to see it spell out IBM. So this was people working at IBM who did this. And here's another one which you can't see. Here they made a little stick figure. Each of these little blobs is a single atom. Now, you can actually do interesting things scientifically. So in these images, here again you can see the same kind of notion. This is looking at a copper surface, and each of those blobs is an iron atom. And they're pushing them in to build up this circle, right? And you can see them taking pictures of it as they build the circle up. And interestingly, once you finish, you made this perfect circle, you'll see these little ripples inside. Now, actually what that is, that's not a feature of the surface. What you're seeing here is the fact that electrons, in addition to being particles, are also waves. And what you are seeing here is the wave nature of the electrons. These are electrons that are bouncing around inside this circle, and they're generating interference effects. So those waves are the ripples in the probability of finding an electron. So you're actually imaging the wave function of the electron as it happens. So you can make different shapes of structures. Here's a hexagon. Here's a circle with two rings of atoms. Because as it happens, the electron waves can sort of leak out between the atoms. But if you put two rings, then it makes it stronger. And in fact, those ripples are deeper than you saw in the previous image. And you get a very different pattern in the hexagon, here in the triangle, and here's a square. So you can make all sorts of structures of this. Again, this is constructed at the atomic level, right? You're picking up and moving atoms. This is a beautiful example. What they did was they made, instead of a circle, they made an ellipse. And you may remember that one of the mathematical properties of an ellipse is that it's got two foci. So if I make a wave, if I draw a ray coming out of one of the foci and bounce it off any wall of the ellipse, they all go back to the other focus. So in this case, they placed an atom right here, it's in blue, at one of the foci, and you can see that even though there is no other atom at the other focus point, which is right there, you actually see some electron density there. So what's happening is that the waves coming out of that atom are bouncing off the walls and converging at that other point and generating a ghost atom. And here they did the same thing with a stretched out ellipse, and so the points are further apart. And just to show that if you place it not at the focal point, you don't get this effect. So here they've got it off axis, and you don't see the image. And again, here you don't see that ghost atom. So there's interesting games you can play with this and do some very nice fundamental science. Now, this is, a remarkable achievement, a very fundamental step, which is direct mechanical manipulation of individual atoms. However, it's very, very hard to do. There's a small number of labs in the world that can do this. You have to do it with an ultra-stable instrument, any kind of vibrations, and of course, if I'm trying to position an atom, it gets messed up. It has to be done at just a few degrees above absolute zero, so very low temperature. You're quite constrained in the choice of materials you can do it with. In fact, there's a very few that this will work with. It's highly error-prone, so you make a lot of mistakes when doing this. And of course, it's not going to scale up to any kind of industrial process, right? This is not a self-assembling thing. You have to move every atom yourself, and it takes hours to put together one of those. So obviously, you're not going to make trillions and trillions of copies of these. So it's not routine, even after 20 years of research. However, there's something else that you can do with this, which has some interesting implications for computation that I wanted to talk about. And this is work that's actually going on right here at the University of Alberta. And so what they're doing is, instead of moving atoms, they're going back to silicon surface. So this is the silicon surface, and each of these blobs is a pair of atoms. And you can actually you can remove electrons or remove atoms and generate what's called a dangling bond. So you get an unpaired electron. So you can bring your tip in, zap the surface, and you get this dangling bond there. So you've removed a hydrogen atom. And you can place these wherever you want. So here's an example of a bunch of single dangling bonds. They end up showing as really bright spots because there's electron density there, and that's what you're sensitive to. And the cool thing is that once you've got these, they're very, very reactive. So you can place these where you want and then throw in some molecule which, say, will react with that dangling bond, and then you'll create a pattern of atoms on your surface. And they've done some work showing that you can actually generate wires that will grow. So the growth will sort of catalyze itself. So you can just start off the wire and then it'll grow itself, which is kind of cool. But you can also actually put a few of these bonds together and watch them interact. So here they placed 4 of them together. So you get these 4 blobs like that. And as it happens, if you've got bonds near each other, they start to interact, and there's some interesting things that come out of this interaction. They effectively tilt the occupancy. So instead of having the same amount of electrons on each of those 4 sites, you end up, right, if I put, say, some charges at the corner here, I can make it so that I'm more likely to have electrons there and less likely to have them there. So nearby charges will change what I'm seeing. And so they can do this in real life, and that's what they've got here. You can see these blobs are bigger. They're not a huge amount bigger, but they're bigger. They're detectably bigger. And this is very interesting because you can use it for the basis of what's called a cellular automaton. What this is, it's basically just a different way of doing computation. So you can think of it as a grid of cells, each with discrete states. So this could be, say, 0 or 1, on or off, right, like a binary system. And the state of the cell is set by some computational algorithm by looking at what its neighbors are doing. And the algorithm is basically the important property in this thing. So what I'm showing here is actually a movie of this it's called the game of life. It was built in the 70s, I guess. And it's just exactly this kind of thing. There's a particular algorithm that tells me, well, if my neighbor is on, or the sum of my neighbors is 1, or you can do it in any way you like, patterning it based on what your neighbors are doing. And you can generate very complicated behavior out of incredibly simple rules. So in this example, right, you see this pattern that sort of propagates itself, and then you generate these sliding lines. And you can get some stuff, I mean, none of that was designed into it. They sort of made this algorithm and then find that it made some very interesting behaviors, patterns. So you've got these, they're called sliders, that sort of shoot out a series of little birds flying along there. Now, you can do the same thing with these quartets of charges that you've built into your silicon. In the sense that you end up with 2 that are more occupied than the others, right? So you could have it like this, or you could have it tilted the other way. So now I've got a binary state. It's either 0 or 1. And the nice thing about this is that, unlike regular computation, it requires very little energy to do anything with this. It takes a fair amount of energy to build it in the first place, but changing the states is not too difficult. And people have worked out how you can make all sorts of do all sorts of functions with this. For example, if I just string a bunch of them together, they act like a wire. They transmit that state down a line. I can make an inverter, so I flip a 1 into a 0. I can take an input and generate 2 outputs. You can actually build every logic gate that's available in digital logic you can build with these cellular automata. And in fact, quite recently, this same group had a bit of a breakthrough, and they can now, instead of just making 4 of those, they can write hundreds of these. There's still some issues with error correction and stuff, but this is doable. It's being done right now. Here's a movie that sort of illustrates what's going on. Better play it. So this is the quartet, right? We've got 4 seats, and effectively, I'm looking at where is the electron density? So these guys are the electron density. And they repel each other because they're the same charge, so they want to sit opposite each other. And if one of them changes the seat, then the other one has to also. Then if I put them next to each other, they'll communicate that same interaction down the line, so each one wants to align up. If I change this first one, I'll propagate that down the line. Then if I bring in some other external charge, like say an electrode, I can fix it, right? And I can force everything to be the same. And then so this is changing my input. And then it propagates down that line. Then of course you can have a whole lot of these, and you need lots of them in order to build these digital logic gates. But the notion is that this involves very, very much less energy than standard computation. And of course, for doing something on an incredibly small scale, as we talked about yesterday, heat dissipation is a very important problem. So in fact, this allows you to scale things down enormously, because now my structures are being built at the level of single atoms, right, as opposed to 100 nanometers or so. So that's angstroms rather than nanometers, hundreds of nanometers. And we've already built in the solution to the energy. So there's actually a company that's working to try and commercialize this. All right. So moving on to the second topic, this is biology as a template for nanotechnology. And there's basically the idea is that you can hijack the great properties of biological systems in terms of self-assembly and reproducibility and error correction and stuff like that to generate structures with particular functions that work at the nanoscale. And one very nice example of this is known as DNA origami. So this was invented just last decade, I guess. And it's based on this notion that DNA, right, is held together. DNA is held together in a double helix. Here's a crystal structure of a little DNA helix. It's held together by these base pairs. It's got a very simple language. The energies of these are really very strong interactions. So you've got a strong driving force that should give you the correct thing. If you remember from last class, I said one of the ways to make things work out right is to have a strong driving force that makes it be right the first time. And you can basically predict what kind of structures you can get if I have a whole bunch of these little pieces of DNA that should base pair in a particular pattern. This should generate structure helices that you can cross-link, and you could generate a geometry that you want. So here's some examples from this really seminal paper back in 2006, where they generated just like a little mat, a star, a smiley face, various triangles. This one has some interesting textured parts in it. So this is what you're supposed to see. This is what you actually see. These are electron micrographs of what they found. And you can see that there's, in fact, many copies of the smiley faces, right? It works. You can do this. Now, these are relatively small structures. You can build slightly more complicated patterns. You can also, one of the nice things about DNA is that you can functionalize it chemically. So in this case, you can put little shiny particles onto it. So they've made this mat that spells out DNA, and then you can see the double helix of the DNA. They've made a map of the Americas here, and then you can tile that. So you can make all sorts of patterns. Here what they've done is they've put little hooks on different parts of this molecule. So you can generate this structure, and then this structure will self-assemble into larger structures, so you'll get these bigger patterns or other patterns. You can see it works on quite a large scale. A more practical, I mean, that's very pretty but not terribly practical. A more practical application is someone built a box, right? So this is now a three-dimensional shape. It's slightly hard to see, and these are electron micrographs. It's slightly hard to see them, but you can sort of see the outline of the box. And this is the structure that they reconstructed from making a whole lot of these images and looking at what they give you as a three-dimensional structure. And in fact, it has a lid, so there's an open side here. There's a lid that you can tie down with one extra piece of DNA. So you can lock it down with a little piece of DNA and unlock it. So the notion is, of course, that this is quite small, right? It's only 40 nanometers. You could put some drugs in there. You could functionalize the outside so it targets a particular cell, and then you could make it so that the lid opens up when it gets to the right cell, right, when it hits the target, and it delivers the drug to the target. You can actually make much larger structures. So here you can see some of these. This is kind of thinking about it, or DNA origami like Meccano or Tinker Toy sets, right? So you can see here various beams and things that crosses or sliding channels. And here they've actually strung a whole bunch of those together. So here are the pictures of the actual structures that they get in the electron micrograph. Here they've taken a beam and made joints. So it makes this icosahedron, a 20-sided figure. And there's what it looks like in real life. So you can make these actually much more complicated structures. It all self-assembles. So it's got the properties that you want, because it'll make its own versions, right? You don't have to go in and make each one. That's still sort of on the toy level. So the question is, what can we actually do with this that's really useful? And here's a very recent paper. This just came out a few months ago, where they built, effectively, it's an artificial membrane pore. So it consists of a bunch of DNA tubes that are functionalized, so they're hydrophobic, so they'll insert themselves into the membrane, and then the superstructure that sort of grabs on to the lipid membrane. And now this acts just like any old pore in a membrane. So in fact, and I guess here you can see here pictures of it, right? There's the piece that sticks through the membrane. This is the sheath that clamps onto the outside. And here's a cell that's got a whole lot of them assembled around it. So each of those little towers that looks like a skyscraper, that's one of these pores. And it has many possible applications. So you can think of this as an antibiotic, right? You could design one so that it punctures through the membrane of a bacterium and lyses it, right? So same way that Staph aureus kills cells by puncturing it and causing the contents of the cell to leak out. You can, in fact, people are working on technologies that use nanopores to try and sequence DNA really rapidly and cheaply. So as it turns out, this kind of thing might solve some of the technical problems that have plagued that approach. There's also a huge number of nanomachines or biological nanomachines that are related to ion fluxes, like ion pumps and that rotary motor that I talked about earlier that generates ATP. In principle, you could take this and build this into some kind of artificial machine that works on ion currents across a membrane. So this DNA origami is really a very powerful method. You can build incredibly complicated structures with interesting and useful functions, very simple ingredients, right, which you can synthesize or just really make up very easily by PCR. It self-assembles, so you get very high throughput, large quantities. There are, however, some limitations. So there's a limited repertoire of materials, right? We can do it with nucleic acids. Now, you could imagine doing it maybe with protein nucleic acids or with RNA versus DNA, but it all comes back to that base pairing interaction. And there's only so many things you can functionalize DNA with. There's also a limited range of environments this works in, so it's great for anything that needs to be biocompatible. But of course, DNA helices come apart when you boil them. So we're not going to be able to make some machine that's able to survive a temperature above 100 degrees Celsius. They also have problems with various levels of acidity and other things like that. So again, there's a limited range of environments. And finally, there is no error correction in this. So it turns out that it's not quite as simple as it sounds, right? You have to find the right conditions for it to self-assemble. And that ends up taking most of the time, is trying to figure out how you can get it to work. And that's still not entirely clear, though there's been some very, very recent discoveries, like just published last month, that address how this may work a little bit better. However, it's a very promising way forward, especially for medical applications because of the biocompatibility of DNA. Now, another thing we talked about is biology as a template for machines, right? And so one thing you need when you're building machines is you need motors, things that will cause stuff to move. And there are many examples of motors in biology. They move, so motor proteins like kinesin move cargoes around inside the cell. This is what the kinesin protein looks like. It's got these two little modules called heads, though you walk on them like feet. They use ATP to move. You can see in this movie, hopefully, again, it does just not show in this lighting, but you see this red circle here? There's a little spot in the middle of that red circle. Now, as I play that movie, that's a tiny lipid vesicle that's being carried along an almost invisible wire of microtubule. And you can see that white dot move, hopefully. I'm not sure if it's visible to you back there. It's very faint. They're just at the resolution of the microscope because they're so small. At any rate, there's another motor that walks along. It's a different filament, the actin filament, so this is called myosin 5. It actually walks in very large steps, so you can see the individual steps it walks on. And in fact, in this paper, these researchers were able to investigate the mechanism by which it walks. They were sort of wondering, does it inchworm, where you take one step, your foot goes like this, or does it walk hand over hand like we do, though of course I'm walking on my feet rather than my hands. And so they were able to show that it's a hand over hand motion. In fact, a lot of this research into biological motors is trying to figure out how do they work, because if we want to design motors that do things differently from biological systems, then we need to know the design principles. Now, the kinesin, that one that I was trying to show you earlier that it just didn't show up on the video, it also works in a hand-over-hand method, but it takes very much smaller steps. So instead of 36 nanometers, which is still pretty small, they take 8 nanometer steps. So it's quite a bit smaller. It makes it much harder to see, but even here you can see those little 8 nanometer steps, right? Each of those is one step. So that's one of these heads moving forwards. Another example of a linear motor, so a motor that goes along a linear track, is our muscles, right? How is it that we're able to move around? And muscles, sort of at their root, they consist of these fibers called sarcomeres, which have fibers of myosin. So this is the motor. So there's a whole lot of these little motors. You can see each of these little heads of the myosin. It's a one-headed motor. And then these motors interact with actin filaments. And the whole thing is held together by, there's an elastic molecule called titin, which gives muscle its elasticity, allows it to spring back. And the way that this works, so there's been a lot of investigation over the last 20 years into how myosin works, is that this head moves back and forth. So it pulls the actin in. And that's actually why our muscles can only contract. They can't actually push. And that's sort of illustrated in this movie here that actually shows all the molecular constituents that come in and get hydrolyzed. You bring in ATP, you hydrolyze it, little bits and pieces come off, and the whole mechanochemical cycle of this, so how the mechanics and the chemistry fits together has been figured out. So we know how these things work. Now, of course, in real life, we don't just have linear motors, we also have rotary motors. In fact, we use rotary motors a lot more than we use linear motors. There are some lovely examples of rotary motors to be found in nature also. So I talked about this ATP synthase molecule the other day as an example of an incredibly efficient motor. So it's driven by proton gradients. They turn this crank, and that causes these subunits to move around and generate ATP from ADP and phosphate. Actually, Boyer and Walker got the Nobel Prize in '97 for figuring for the mechanism of this. But it's interesting that they didn't get given that prize until in 1997, Kinoshita in Japan, his lab actually showed that it really was a rotary motor by taking one of these units, fixing it down to a glass slide, putting a fluorescent actin fiber on it, and then feeding it ATP, so running it in reverse. And you should be able to see this bar. So that's the bar, that's this fluorescent actin. And so it actually rotates around. Now, that jerkiness is not something to do with the image or something to do with my computer being really slow. That's actually the motor. As it happens, it takes steps, just like those other motors, except it takes rotational steps rather than linear steps. And it turns out that you can plot these steps. So this is number of revolutions, and you'll see there's 3 steps per revolution, and it turns out that it's got a 3-fold symmetry, so it's basically moving by 120 degrees each time. And in subsequent work, they actually showed that there's many sub-steps in this, and they've been, over the last 10 years, they've been piecing together each of the little pieces of this enzyme and how that all plays into how it works. Again, trying to build up a complete picture of the mechanochemistry of this. There's some other rotary motors. So here's the bacterial flagellum, right? That's the little tail on the end of this bacterium there. You can see that it swirls around and propels the bacterium forward. It's actually an incredibly complicated mechanism. So there's the flagellum. It has this assembly, this motor assembly that sits inside the membrane and wall. And the constituents of this have been figured out, and it took about 20 or 25 years for people to actually be able to see the individual steps in this rotation. They've suspected they should be able to see them, but it's very hard to see. And so what they did to do this was they stuck a bacterium down, put a little fluorescent bead on the end of the flagellum, and then watched that move around. So, this is a very small thing, and so of course it's quite pixelated, but you can actually track the center of that motion quite well to within about 5 nanometers. And when you do, and you plot this versus, this is the angle in degrees, you'll see it takes very, very fine steps. So each of these little blobs in here is a step around the circle. So I think it's, I can't, oh there, 26 steps per revolution instead of 3 for that ATP synthase that we looked at. So it's actually a much harder system to study. There's another type of motor that's actually very useful if we're thinking about self-assembly, and that's polymerization-based motors. So this is a movie of of some cells, these green bodies here, that are infected by listeria. So this is a bacterium that causes food poisoning. You might remember a bad case of this a few years back in a maple leaf plant. And what listeria does is it actually hijacks the actin-forming enzymes in the cell to generate these tails. You can see these comets of green. Right, so this is a cell that's expressing fluorescent actin. And actin is used, it's part of the cytoskeleton, right? So it's part of what gives the cell its shape. But this is these particles, these bacteria are in fact hijacking the normal pathway of generating these fibers to grow a bunch of actin in behind themselves. And as they grow that, it sort of pushes themselves forward. It sounds kind of strange, but actually that's what's going on. Here's a electron micrograph of this showing one of these so-called actin comets in behind the listerium particle. And actually the reason it does this is because it shoots itself out through the membrane and then into another cell by brute force, basically. And the way this works is it's called a Brownian ratchet, basically. So the notion is that my particle, so it's sort of shown here in this cartoon. My particle is like this little bead here. As this fiber grows, so the particle is bouncing around from Brownian motion, right? It's a small object. It moves a lot due to thermal motion. As it moves away from the fiber, it generates an opportunity for the fiber to grow. If a subunit comes in and joins to the end of the fiber, now my bead can't go back all the way. So I've had some net motion forwards. So basically you're rectifying the motion, right? So you're saying only you're biasing it in favor of fluctuations that move in a particular direction. So it's a very clever, ingenious mechanism because you don't really do anything. You use the fact that it's a small system that's fluctuating due to temperature. There's also some motors have been applied you can think of this as nanoengineering, though it's still very basic. So, for example, people have used microtubules and kinesins to direct cargo to be transported to particular locations. You can lay down tracks of microtubules, and then the kinesin will bring stuff into a central location, for example, so you can concentrate analytes, say. It's a little bit hard to see on here, but there's some very faint white lines, which are microtubules that have been fluorescently labeled. And they're sticky, so they'll stick together. And they've carpeted the bottom of the microscope slide with kinesin so that it sort of shuttles these things along. And as they collide, they start to make bigger and bigger fibers. So you can see some long fibers here. And then they end up making these little spools. This is this notion of self-directed assembly of molecules, where you're using these motors to do it. And in fact, other people are working to try and engineer entirely new motors. So this is one idea that's been put forward, which is it's called a tumbleweed, and the notion is that it should move along a track of DNA. So you could imagine making a track of DNA, say, with your synthetic biology or something, with your origami, and then you'd have this thing that rolls along it and gets to where you want it to go. Now, I mentioned synthetic biology. This is another area where biology gives us a template, but we go sort of beyond what biology does to start treating biology more from an engineering standpoint. And it's this fusion of biology and engineering which is really essential to synthetic biology. So you're engineering entirely new biological functions and systems. And it does really go beyond standard genetic engineering, though it has many of the same elements, because you're trying to design things, right? You're not just tinkering with it, you're trying to design things. So it's this notion that you take as a model your natural circuits, you generate synthetic circuits out of that, and then you have mathematical modeling, which allows you to go back and forth, and you can improve your systems by doing this. And in fact, there's a registry of standard biological parts. So you can think about this as the way we deal with, say, electronics. So in electronics, right, you can go out and buy standard transistors and diodes and wires, right, and switches and various types of integrated circuits that have these things put together in particular geometries. These are standardized parts. And then you design a circuit which puts these standardized parts together in a some way to generate a particular function, whether it's, a timing circuit, an oscillator, what have you. And in that same way, they've come up with this idea of, we'll generate standard biological parts with standard connectors so that I can take the DNA pieces that correspond to each of these and bring them together and build them with CAD tools, for example, so you can put these things together and you assemble something that does, so a circuit that does a particular job. And people have been adding, there's several thousand parts on this registry now. So people have been adding, every time you develop a new one, you add it to the registry. And just some examples. So here's an example where they used E. coli that are engineered to be photosensitive and take pictures. So you expose it to an image and it records that image. So there's the masthead of Nature for a paper that was published in Nature. And then here's a picture, I don't know if you can see that, it's somewhat faint, of one of the scientists who did the work. Unfortunately, it's not a great medium in the sense that the visual contrast is not very good. However, as that demonstration of principle, it's quite interesting. Now, more recently, people have gone quite a bit beyond this and started looking at trying to generate entirely artificial organisms. It's not quite entirely artificial, but they took a cell and re-engineered its genome, and they actually put the entire genome together by chemical synthesis. So in some sense, it was trying to prove that you could synthesize an entire genome. Now, it wasn't cheap. I think it was about $40 million to do that, right? So this is not something that you're going to just go out and do right now, but the technology for synthesizing DNA has been advancing by leaps and bounds, and the cost of doing this kind of synthesis is coming down very rapidly. There are, in fact, many, many applications of synthetic biology, many of them very recent, and I'm not really in this field, so I'm not an expert on any of these, but I put some of these up here for you to get a sense of where these applications go, because this is a very incredibly exciting area with lots of opportunities. So one of the applications would be in biosensing. So for example, you could engineer something to be sensitive to a particular reactant. Maybe this is a pollutant, maybe it's a toxin, maybe it's a drug, maybe it's a metabolite in your body, so you can sense the level of metabolites in your body, right, as part of regulating your health. All sorts of things you can imagine doing with biosensing. Another one is producing materials. So this could be, say, more efficient ways of producing biofuels. This could be generating pharmaceuticals, right? You engineer an organism to generate pharmaceuticals. And in fact, there's a commercial venture to produce artemisinin, which is a treatment for malaria, which is currently quite expensive and hard to get. And they'll make it in bacteria. You can also, I think people have engineered bacteria to generate silk, for example, silk proteins. So there's lots of things, any biocompatible material you should be able to generate, or you could, in principle, be able to. You can imagine doing this with synthetic biology. There are people working on digital computation done genetically inside an engineered organism. So this would be, say, using protein expression or RNA expression as inputs into logic gates. So you can have an organism that does a particular computation. Turning to medical applications, you can actually, so one of the interesting ways of using synthetic biology is as a platform for understanding what's going on in a disease, because you could engineer a much more simple system, right, and have a look at what's going on in there. And this gives you an idea of how the different parts interact to make the whole disease. Once obviously this is a long-term effort, but it gives you a way forward that's quite interesting in terms of its potential implications. You can also, people have done this, they've engineered viruses as therapeutic agents, for example, a bacteriophage to go after drug-resistant bacteria. Right? Drug resistance is a growing problem for many bacteria. This is a way around that. They've also come up with ways of increasing antibiotic efficiency, for example. There are tumor invading, so some bacteria have been engineered now to invade tumors. So this is a potential treatment for cancer. For example, they could deliver drugs but right to the tumor. And then there's even some demonstrations of how you might be able to modify disease vectors. So there's a scheme to generate flightless, to infect mosquitoes to make the females flightless. Again, not all the way there, but some very interesting first steps taken on that road. I think one of the key things to keep in mind here, though, is that just as genomics ended up being a lot more complicated than we thought, right? We thought, well, once we've got the genome, we should understand everything. But as this person said, genome, bought the book, hard to read, right? Hard to figure out what it tells you, what it means. Someone came up with a similar statement for synthetic biology. Synthesis can write DNA, little to say, by which they mean it's actually a lot trickier to make this work than you would think. So the syntax of the language is not easy. Now, finally, I'd like to talk a little bit about nanotechnology and safety. So gray goo really isn't what we should be worrying about. We're nowhere near there yet. Although, as we talked a little bit last class, self-replicating organisms really are a source of worry, but they're not really much of a different source of worry than we already have from current technologies, biotechnologies, genetic engineering. We can already make really virulent viruses, and that's as much of a problem as anything you could generate from synthetic biology. I wanted instead to concentrate on something a lot more prosaic, which is this notion of toxicology. So there are already on the market very many products that use little nanoparticles. Whether this is in sunscreen or antibiotic, silver nanoparticle antibiotic treatments in Band-Aids, or various other particles as coloration agents. They're out there in the market already. And this question actually ends up showing you some of the problems with how to deal with, some of the problems that arise when you start talking about what can we do to improve safety around nanotechnology. And the issue is it's not just a question of what materials you're using, right? When I buy a jar of toluene, right, someone has tested the toxicity of this toluene, right? It doesn't matter how much of it I've got, in a sense. The toxicity isn't a function of how much is there. Take the example of zinc oxide nanoparticles. So these are found in sunscreen. Zinc oxide in bulk is a perfectly harmless substance. There's no problem with it. But as it happens, it has toxicology. It's toxic at different amounts for different sizes and shapes of particle. And that's a real problem, right? Another example is carbon nanotubes. So carbon, of course, is a non-toxic material. But if you turn it into long fibers, it turns out that the shape is what's toxic, not the material. So just like asbestos fibers will generate asbestosis, you can get something similar from certain types of nanotubes. But they have to be the right type. Not every nanotube will generate this response. So it has to do with the shape, not the material. Now, people have also shown, for example, that nanoparticles can partially denature proteins, and since our proteins require their structure in order to function properly, if you're denaturing them, that could generate dysfunction in cells. So here you can see various denaturing for different types of proteins. It turns out that they will also, so not just mess up proteins, but they can mess up the membrane, the cell membrane. And since that's sort of what defines the integrity of our cells, anything that changes that will have some important effects. So you can, in fact, change the curvature of these things. And that is not necessarily, that can do bad things to cells. And people have looked at, you can look through this after the class, people have looked at various possible places that nanoparticles might end up causing problems. So possible toxic issues and diseases that these might be connected with. So I think this sort of highlights some of the uncertainties in dealing with even something as simple as, is this substance toxic or not, right? So there's a rather fundamental question, which is, what is the important feature, right? Is it a dose by mass? Is it the surface area? Because the surfaces that end up being quite reactive in many of these things. How are we going to evaluate the toxicity? There's a question of, how do these properties change with time? So many small things, for example, will over time tend to aggregate into larger bodies. Do they become less toxic as they do so? So does this mean that we should worry about that the toxicity starts off strong and then becomes smaller, or does it become more toxic as it aggregates? And we have to worry about something that's not toxic at first and then becomes toxic. There's also the question of where these things end up. So this often hasn't been looked at, right? Do these end up, when we put one of these on our skin, where do those little nanoparticles end up? Do they get absorbed into our bodies and then excreted? Do they slough off with the skin particles and then end up in the environment and then get washed out into the ocean? And then what happens with them after that? So there's this question of, what's the life cycle of these particles, and what do they do at each point in that life cycle? Then there's this question of when we're looking at different environments, say in water versus in air versus contact to your skin versus inside your body, what's the important level of exposure? And finally, this is sort of the most basic of all, is there any aspect of the toxicology that's predictable from the bulk material? So if I take something that's perfectly harmless like carbon, which you're largely made up of carbon and water, and I spin it into a fiber and all of a sudden it becomes toxic. If there's no way of predicting under what circumstances a material will become toxic, this is sort of a nightmare, because it means that any time you do anything, you change the size even, or you change the shape of something that's very well characterized in other circumstances, then you have to worry about, have the toxic properties changed? So we would need some way of modeling this, right? And just to end off with a cautionary tale about regulation of things, of course, is that it's always a question of framing risks and rewards appropriately. So this sign says, hikers and bikers move to the side of the road when a vehicle approaches, and you can see a whole lot of alligators coming out of the swamp. Right? So it's a question of, are you aware of all the risks that might be there, and are you balancing them appropriately versus the reward that you get? And with that, I'd like to thank some of the people whose ideas and materials went into the lectures over the last couple of days. Steve Blocka is a professor at Stanford University. Robert Wilkel is a professor here at U of A. Larry Sharameta, who's also at U of A, and then Paul Burrows at Riata Research. And thanks very much, and I'll take your questions.
Hi. I just had a few questions. You were talking about the different temperatures that a molecule, when you affect a molecule, or not a molecule, an atom rather, then the temperature state has to be a couple of degrees below zero, I think you said, or a couple of degrees above. I was just wondering if that temperature was relative to the type of the molecule or the atom.
So first of all, that's absolute zero, right? So that's minus, you're doing this typically down at minus 270 roughly degrees Celsius.
Okay.
It's not zero as in freezing, it's absolute zero temperature.
Okay.
So it's very close. And yes, it does depend, like the details of that do depend on what surfaces you're looking at and what atoms you're moving around on them.
Okay. And I was also wondering, when you were talking about DNA origami, from my clinical agenda, there's a lot of cardiac diseases such as myopathies that, one of the reasons they lead to sudden death is because of changes in potassium pumps and calcium channels and that sort of thing. Is that the sort of agenda which this can be applied to in order, can you create a DNA channel which, moves potassium or calcium selectively through, sort of bypassing the problem?
So that research I showed you was just published a few months ago, and I'm sure people are still digesting what the implications are, but I'm sure that's certainly on the list of things that people are thinking about, right?
Okay.
There's always practical issues about how you go from the first demonstrations to something that's, actually in your hands to use as a clinician. So I'm not sure how long that would take, if it ever happens.
Okay.
But I'm sure it's something that people are looking at.
So because I don't work with molecular or basic sciences either, so I'm not sure of how, at what point, at what stage it becomes, it enters the clinical realm. Do you have any idea?
Yeah.
I mean, if you're talking about pharmacotherapy, you're looking 10 to 15 years from conception of the idea to production.
Certainly one issue here is that the channels they made are not ion selective. So there's no ion selectivity built into it. So then you would have to figure out, how can I make it ion selective, for example, to do this kind of thing. Right now it just makes a pore.
Yep.
And so it would work as an antibiotic, so it would lyse the cell. But that's not really what you want.
No. Understood. Thanks very much.
Relating to safety, I was just wondering if there's ways of detecting the nanoparticles or monitoring them in any way, like outside of a lab potentially, or is that still a major obstacle?
I think this is a difficult prospect currently. So I don't, again, that's not really an area in which I specialize. And I know there are people thinking about how you do this, but I'm not actually sure how far they've gotten. I don't think it's easy at all, because you've got to separate out these tiny little particles from everything else.
So aren't we dealing with something where to evaluate all cases is just absolutely impossible?
Yes.
So, you can take substances used in a certain way and test them all in animals and that sort of thing. That's just one level. But suppose that the toxicity depends upon the concentration and a bunch of other things, and the shape. So it's not just how much you have, but you end up with so many different possibilities that you could never justify testing for them all.
Well, in fact, as it turns out, I think the majority of chemical compounds that have been made by humans, we don't really have any toxicology data for them, right? Because exactly this reason.
Yeah.
For stuff that's used very rarely and in very limited circumstances, there's just no call for doing it. And obviously, that's the kind of approach we should be taking, in the sense that presumably if you're making something for, say, use in or on your body, you might want to know a lot more about its toxic properties than some material that you're just making in the lab to look at some particular, maybe it's a reagent that goes into the early steps of one of those other things that we're talking about, but it doesn't come out of the lab. Then obviously you wouldn't be so concerned about the toxic properties. And there are other issues too. You could imagine that we now know enough to say that most things that end up with the shape of an asbestos fiber will probably be toxic just because of their shape, and it wouldn't matter what they're made of. Mm-hmm. So then the presumption would be, well, if you want to make something that's like that and put it in a product, say, that goes on your body, then you should show that it's not toxic, right? Because the presumption then would be that shape is itself toxic.
So it becomes a kind of scientific challenge in itself to find a way of testing toxicity that doesn't involve animals, right? And there's one bit of research that would indicate the amount of animal testing is maybe going to go up 1,000,000-fold, and another bit saying, no, we have to bring it down, right?
Well, and certainly I think one of the ways forward is, I sort of hinted at this at the last slide, but this is modeling, right? As
Yes.
we develop better models of toxicity, we'll be able to put in new things that we make into these models and say, okay, is it likely to be toxic or not? And if there's some likelihood that it's going to be toxic, then we could screen stuff out that's not likely to be toxic and just never test that, and just test the ones that are. And as we do more and more testing, obviously you can refine these models. So I think you have to have something like that as the way forward, just because this is the only thing that can scale properly.
Yes. Thinking about the challenge of food and the idea of growing meat in the lab without ever having had an animal that the meat is from. You just decide you want a certain kind of meat, and you can grow that meat without having the animal. It would probably be possible to do testing of toxicity of certain types by just having the cells without having an entire being.
Yeah, well, this would tell you certain types of toxicity, right? But it wouldn't tell you things that, say, for example, you have little particles that are able to cross into the brain and then aggregate there and trigger some response, kind of like Alzheimer's, right? Just as a thought to throw out there. So there's a lot of, I think, organismal level things, so that they're much higher than just what does it do to a single cell.
Yes.
That would need to be, you would want to look at.
Right. Must be other questions.
For these self-assembly things that just keep replicating, like, is there going to be a way to kind of limit it so that, like in biological systems, you got a feedback system so that it limits itself?
So, I mean, there's certainly notions that people play around with. One of them would be to basically have them require some element or some ingredient that just is not in the natural environment. So then it'll only grow when you provide it with the appropriate medium, right? And this is one method that's used currently to, so this is more than just containment. This is basically saying, well, even though I am containing it in a biosafety lab, I've also designed it so that it shouldn't be able to get out. Of course, the worry is that you may end up generating, just by mutations and evolution, you may generate versions, generations of this that don't need that anymore. They've somehow found a way around it. Especially if you provide, right, if it's constantly testing these boundaries and trying to escape, and then it's coming up against this limitation, and those organisms die, but every so often something might, in fact, be able to get over it. So basically, I don't think there's any foolproof ways of doing this.
Great talk. I have a question regarding targeting the cancer cells and delivering the drug with nanoparticles to those ones. We haven't achieved that much, I think. What we were thinking that it will really work, it is not over there yet. You think it
I'm
is?
sorry, is?
The goal is not achieved
Gold,
properly.
yeah.
Do you think it is because of lacking of biological knowledge, or it is nanotechnology?
So you're talking about the work that's been done using gold nanoparticles to try
Exactly.
and target cancer cells.
Yes.
Well, my feeling about that is that some of the difficulty is no doubt the fact that it's not particularly targeted. Right? Some of the strategies that have been proposed are much more selective and could deliver something you have designed to kill that cell. Right? Whereas, I mean, gold is just gold. Right? I'm not actually sure of the mechanism by which it kills the cell. Do they heat it up or something and then it's supposed to harm the cells? I'm not actually sure about the mechanism. I'm sorry?
After the gold part is supposed to start,
Yeah.
apply laser
Okay, so they heat them up. They heat them up. Yeah. So that, I mean, that's a very crude. That's kind of like taking a sledgehammer to things and hoping that you'll break the right thing. Whereas if you could, for example, with these synthetically engineered bacteria that seek out and enter cancer cells, if you had one of those that then contained within it the chemotherapy drug you wanted, you'd now be delivering something that could be very, very, like far too toxic to go into normal cells, but you deliver it specifically to that cancer cell. You know, that would be a lot more specific way of killing it than just trying to heat it up. But I agree, there's a lot of work to do on all of these methods to make them actually practical in the clinical setting.
It's not a question, but I cannot imagine like how the carbon itself is not toxic, as you told. And then when it starts changes to be like tubes or something like this, it's toxic. I cannot imagine this poisonous. I cannot.
So as it happens, they've done the same like toxicology studies with even carbon nanotubes of different size, like length of tube and different width of tube. And only some of those sizes are toxic, and the others are perfectly fine.
I cannot imagine it, but it's the same particle, the same material, so yeah.
So the issue seems to be that it's not the material, like it's not asbestos that causes asbestosis, it's actually the shape. The shape is toxic.
Yeah, maybe. Okay.
Yeah, it is, you know, it's a different, you
Yeah.
sort of maybe have to change the way we think about what's
Yeah.
generating toxicity.
Okay, thank you.
You touched on this lecture as well as the previous one, the ATP synthesis motor. I was just wondering if you know why it's so efficient. Is it just there's a lot less friction, and also if people have attempted to replicate it at a larger scale?
So that same enzyme is found in basically almost all organisms. And if you think about it, it's so absolutely central to life that any tiny improvement on it is going to improve the efficiency, the survivability of an organism, right? So any organism that has had a mutation that made it slightly better, even like half a percentage point or a tiny fraction of a percentage point, that gives it, you know, a fitness advantage, and that'll be selected by evolution, and evolution's been working on this for a few billion years. So that's why it's so efficient, because it's just so absolutely basic.
Okay, I was just wondering, like, as opposed to you last class talked about like a car engine only being about 40% efficient. If I know like a car like loses energy going through transmission and heat and friction, blah blah blah, do you know exactly why it's?
Well, I mean, there's other biological motors that are nowhere near so efficient, right? But if you think about it, the purpose of that enzyme is to basically generate an energy currency. So all it's really dealing with is energy. So it had better be as efficient as possible. Many other things have other competing interests, right? Maybe you need to make sure that, I would say in the case of a motor enzyme that walks along, like kinesin that walks along a track, maybe you have to also be careful to make sure that it can take enough steps in sequence before it falls off, right? And then you're evolving it to try and do that as well as be efficient, and there's trade-offs between the two. So when there's competing pressures, you're not going to get quite as good in either area. It's just like, this is, you know, there's design trade-offs in our engineering. Typically, we have to make compromises about how something is built just because we have competing interests that we want it to be able to do two things, but to optimize it in one means to degrade it in the other, right? So you end up with a compromise.
Okay, thanks.
That would be my guess, right? Obviously, I don't really know for certain, but that would be my guess.
Yeah, and you don't know of anyone who's, because it looked like from the images that we know how it works, basically, kind of. So if we can try and make something similar, I don't know.
When I say we know how it works, I guess what I'm really trying to say, we've figured out many of the steps that are involved, or most of the steps that are involved. Now, for us to generate our own motor is a much bigger problem. What people have done is they've taken some of these motors, like myosin, the one that's in your muscle, and they have engineered that, say, it moves in one direction only. They've made a version, say, that moves in a different different direction along the same fiber. They've also made ones that have different step sizes, right? So you can start tinkering with it. So we're at the tinkering level. You can change the speed at which it works, for example. But I think we're, the efforts, I showed you that, that one effort at trying to design a motor from scratch based on biological principles. That's still a very simple thing, much simpler than any of the motors that evolution has provided us, right? And that's because we haven't had a few billion years to work on things yet.
Okay, thanks.
So kind of basing off what she was talking about and how you mentioned they have kind of nanoparticles or nanoassemblers have the safeguards and their self-replication so that, they're not just everywhere. Is it feasible that if someone wanted to and had the technology and the mental know-how, is it feasible to create a self-assembler, an assembler that could not be stopped from assembling things? Then could it be feasible to make an unstoppable gray goober?
Well, let me put it this way. It's certainly feasible. People have done it. You can make an incredibly virulent virus that, if it got out, it would infect, a large fraction of the population. And this is an incredible, that's a very, very efficient self-replicator, right? We can do this already. We don't need any new technologies to do that. We've been able to do this for a while. I mean, fortunately, as it happens, most of the people who have the technical ability to do this are not evil, right? It's not a James Bond movie.
Thanks.
So other questions? The synthetic biology talk, Hacking Life, is a week from today at 4 p.m. and I think CCIS L2-190, Andrew Hessel. And I think you'd find it quite interesting if you happen to be free at that time. Very interesting young guy associated with Singularity University. Other questions? We've still got 2 minutes. Anything else? Going. Okay, well, thank you very, very much.